I think we can get started. So welcome everyone. Today we're going to have a focus group where we'd love to get your opinions and your feedback about tools that we're offering in the knowledge portals. And I'll go over some existing tools that you can use and that you may not be completely familiar with, and I'd like to get your feedback on those. I also want to hear if there are just things that you would like to do with the knowledge portals that you can't do, or visualizations that things you can't see that you would like to see. We'd love to have just general feedback. And then we're going to talk about some in development tools. But right now, Laura Scott from the University of Michigan is going to present work that she and her collaborators have been doing on a new in development tool. We're just doing this first because of scheduling constraints, but Laura will talk about this first, then we'll go back to some of our existing tools, and then at the end, I'll talk about some other tools that we've got in development. So go ahead, Laura.
Thanks very much, Maria. Thanks, everyone, for being here. I'm curious, I did this back in July, and just to get a read, can I see like a thumbs up from or some sort of indication on your screens whether you've heard about Colocus before, just so I kind of have a sense who I'm starting with. If you find that little button, I'm not seeing much. Okay, so I will assume little background on this. So what I want to tell you today briefly about is a tool that we've been developing. And by we, I mean Ryan Welch, Jeff Holtzman, and Mike Binke and I, called Colocus. And it's a molecular trait browser specifically to look at colocalized signals. So the goals of my presentation are the rationale, an introduction, and then to provide you an ability to actually use the tool, because we would love to get feedback from you. And I just want to point out that this is a tool in development, and so there may be bugs in the staging version. Okay, so why do we want to colocalize things? So what you see here on the top is a GWAS for waist-hip ratio adjusted for BMI, the chromosome position X, log 10 p-value, and you can see a GWAS signal, nice strong signal, single variant up here in the lead. And if you look at the gene, so if we've done an EQTL study in subcutaneous adipose, and if you look at the gene CBX3, you see what looks like a very similar pattern. So if you were going to make a guess about a gene that might underlie this GWAS signal, this looks like one that might be plausible from this overlap. And so what we want to do is to give a tool for you to assess kind of how good overlaps look, how many genes look like they have the same overlap. And so we want to identify genes, metabolites that may be causal for CMD traits, identify sets of CMD traits that may have the same genetic underpinning. And we want something that is going to play well with other browsers, because it's going to be part of the knowledge portal and potentially other browsers. So things that we want to know, and things why we're building this, is how much should this colocalization of this gene with this GWAS, or lack thereof, increase, decrease my belief? How much should I believe the genetically predicted direction of effect between level of a gene, protein metabolite, and disease risk? Okay, so I'm going to show you two different ways to get to this tool. So there is right now a version of the tool running on the knowledge portal, and that version is under the KP Labs, and it currently has the Adipo Express data, which is a subcutaneous meta-analysis. That the underlying data and the underlying tool will be kind of continually updated as we have production releases. But at the next CMD-KP release, which will be between March and June of this year, Colocus will move from KP Labs, where you can find it now, to tools. And we're also going to start to include, when you search on a gene in the portal, you will have a Colocus view that you'll be able to click on, so that will be integrated into the portal, which is one of our strong goals going forward. Okay, so the current data in Colocus, it's an AdipoExpress meta-analysis, about 2,000 people, almost completely European ancestry. We've got a paper on bioarchives. Right now, this is done with Karen Mulkey, Corinne Small, and I. So that's all the data that you're going to see today. So the things that I want to show you today, these are features. We want to browse for colocalized GWAS molecular trait signals, allow us to assess the quality of them, visualize multiple signals in a region, and be able to browse that data. We have other things that we're going to add as we go along, but those are the ones that I want to show you today. So the first views I'm going to show you are the current production values that's in the KP tools on the portal. So this is what the screen, approximately what the screen looks like with some changes. It looks like when you go in and you press on Colocus in the KP tools. So you'll see there are two parts to this. There's a side with a menu and then a side with results displayed. So I'm going to start with the menu. So in the menu, you can put in a gene name, a genomic region, GWAS phenotype. You can click on that. Your favorite colocalization parameters, with 1 being the highest, 0 being the lowest, 0.5 or 0.8 are things that people use. You can also use an R² between the lead SNPs from the two different signals. People like to use sometimes 0.5 because you can get colocalizations where the R² is not actually that high between the two lead SNPs. And then you could say, I want to make sure that the p-values or the negative log 10 p-values are above some threshold, because you can get colocalization of signals that don't have such strong things. So we've set some defaults, but you can set those yourself. Next, we've got at this point in the browser only GWAS data. So we have the trait 1, which over here is the GWAS. Then we have the trait 2, which is the gene, the tissue it comes from, color codingness for the variants, which we've done in a couple different ways. But basically what this allows your eye to do is, these are random colors, but if they match, right, between SNPs, either for the same trait or between traits, they will be the same color. So very quickly you could say, this is a signal that both of the lead variants in the signal, the conditional signals, match each other. Okay, they could be in high LD, so they don't have to match. You can see that here's a set down here where you've got multiple genes that have the same multiple genes colocalized to a GWAS signal that have the same variants. Here you can see the p-values, and these p-values are for the conditional signals. So we have done, we've used Apex, people often use SUSY to isolate and find map signals. So these are not the marginal signals. These are actually the signals of the leads that we're colocalizing. And then here you can see that H4, the colocalization, so these are really high, and then the R². So here's an instance, it's the same variant. So R² is 1 between the two traits, colocalization of 1. So this is strongly colocalized. We've replaced this signal, we're replacing it with something more intuitive, which I'll show you. Okay, so here's an example where if you look at the browser, you can see there's the same trait, many different genes. This is a beautiful set where you've got strong evidence of colocalization for many different genes with the same GWAS signal. Okay, so now I'm going to switch to the staging browser for the AdipoExpress data. And I think, Maria, you have or are going to, in the chat, put in this URL so that you could actually do this at home if you wanted to.
Yes, the link's in the chat.
Beautiful. So this is the same starting browser I had before, except we've switched now to just color coding the SNPs in colors instead of having big boxes. We've added two things. So this is the number of colocalizations with trait 1 and trait 2 pairs. So you can see that for this pair of the trait here, which you can't see, it's a waist-hip ratio adjusted for BMI, and this gene, there is only one set of colocalizations. Here is a gene where you have 3. So this one has what looks like it may be an allelic series. Here you can see whether the direction of effect between the GWAS trait and the gene are concordant or not. So here, dis means opposite, and then concordant means concordant with each other. So it's just whether they're positively or negatively associated if you're interested in kind of what it might say about the effect of that gene on the trait. Okay, so I want to show you an example today for RSP03. And you can see, even though you can't see the header here, you can see that this gene has 2 different signals, right, between the trait and the gene. So to get to this row, you can do 2 things. One, you can click on the row, or you can put RPS03 into the EQTL box. So I'm going to click on the row. All right, and that brings you to what we call the locus zoom page. So on the locus zoom page, you can see that this is the signal we selected. So you see on the left, let me back up actually for a minute. And you can see that there's an LD, there's a box here. So this box allows you to switch between seeing the conditional and the marginal signal. And it also allows you to switch between whether you want the lead variant, which you can see the color coding of this displayed, or the lead variant with the color coding of LD of this displayed. You can see the locus compare plot between the EQTL and the GWAS here. This looks like a beautiful colocalization of the strength of the signals. And then you see the locus zoom plots between those 2. All right, so let's say that what I want to do is I want to actually look at the other signal. So when you want to look at another signal, you click on the row for that signal. Let me just make sure I got this right. All right, so let's say I click on the row here for this signal. And now what's happened, you can see it's highlighted in blue, so it puts up the signal for the second signal. So the locus compare plot that you see here corresponds to the second signal. Okay, so now I've been able to see what both of the signals look like. They look fairly good, fairly convincing. You can see there's a fairly strong signal for each. Those signals look very similar. Okay, so now I'm going to jump back to the first signal, which I can do by clicking on the row for the first signal. Okay, now I'm showing the first signal again. So now I'd like to see the 2 signals together. So I've told you that if you want to see an individual just row, you click on it. But what you can do is to add more plots to the same screen, you can click on the plot icon. So the plot icon is found on the far left. And if you click on the plot icon, what's going to happen is a few different things are going to change in your screen. So one, the top 2 plots and the locus compare are going to stay the same. Okay, but now I've added the signals for the bottom 2, the bottom pair. And so what we have here is the triglycerides meta-analysis of one signal, the triglycerides meta-analysis of the other signal, and then the genes that are co-localized to each of those so that you can see them together. If I want to change the LD here, I could change the LD to be the GWAS, the lead GWAS signal. So they correspond blue, blue to keep you straight. I can change that to correspond to this. So if you watch, the LD is color-coded from kind of purple to lead, red down to blue. So now what's going to happen when I click on this snip, click on change this toggle, is that now I can see the LD of the other signal. So this allows you to go back and forth in one easy way. So now I might also want to see, like, what did the marginal signal, what did the original signal look like in this region, right, for this trait? How different are these? You can see that this signal has a negative log 10 p-value of 40, this one of 50. These are less, this is 15, this is 20, right? So what did it look like to start with? So for that, I can change to the marginal p-value, okay? And now what you see, so I've still got this SNP selected. So the locus compare changes to looking at the marginal signals. And what you can see, so I'm using this as my this signal as my LD signal. So you can see actually that the two signals together look kind of similar, but they're not as well differentiated. If you had tried to just co-localize these two, it wouldn't have been as good, because you can see that there's a pair right here that's not in that kind of is offset here, which are the two different signals. So our goal is to allow you to kind of see, what did the original signal look like? What does the separation of the two signals look like that we have? Okay, and we have a feedback thing. If you'd like to give us feedback, we'd love to have that. We have planned additions, and we're going to add more data in than adipose. And so I hope I've been able to show you today kind of the ability to compare the signals, facilitate searching, and provide a context to colocalization.
Laura, there's a question in the chat. How do you handle add mixed populations in the colocalization analyses?
That is a great question. And I don't know the answer to that. All the stuff that we've done so far has not been admixed. And I have not been following the literature. I guess what I wonder is there's a tension in the world of finding multiple signals between doing a mega analysis and a meta analysis. And I guess by admixed, that could mean two different things. That could mean African Americans, or it could be combined samples of European Americans and African Americans. What did that question mean? Whoever said that, what did you intend by that?
So yeah, I intend both actually. This is Nora.
Yeah.
Thanks a lot. A very nice presentation. So yeah, I mean, we're trying to avoid the use colocalization, unfortunately, which is a fantastic method because we do deal with the mixed populations, and it could be like even if, and you're right, if you meta-analyze multiple African Americans, we cannot go to the local ancestry specifically, right? But it's being an issue that I think someone has to resolve in the colonization. Yeah.
I don't have a good, I mean, we've worked on developing a tool called Apex that allows you to do co-localization of individual studies so they each get their own LD. I think there still could be issues, but I think it's a great question and I don't have a good answer.
I just wonder if we should put a caution for people using this, because I know people will use for whatever they want.
So at this point, we don't have a way for people to load their own data.
Okay.
At the point that we do, I agree completely at the start, a lot of those cautions need to go in. Complete agreement.
Thank you. That's a great tool. I will use it.
Great. We would love to get your feedback. I see we've gone over, Maria. I don't know whether we should ask for more questions or have people write.
That's okay. No, if anyone has any other questions or feedback, this is a good time. Maybe not.
Okay, so please try it out. Can I see how many people actually tried it while I was presenting? Did anybody get a chance to do that? Just curious for other presentations. Okay, so
Okay.
if you do get a chance to do it, good, fantastic. Um, if you do get a chance to do that and you have any suggestions, we would love to hear them. So please click on the feedback link on the staging version. Thanks, Maria.
Thank you, Laura. Okay, let's continue. I'm going to share my screen now. And I'm going to talk a little bit about some existing tools that we have that you may not have really explored fully. And then I will talk about an in-development tool that sort of combines the functionality of both of the tools that I'm going to show. Oh yeah, but first I wanted to ask, actually before I'll stop my share, does anyone have any just random suggestions? I mean, just something like a query you would like to be able to do, but you can't, or some roadblock you've encountered, anything, please feel free to speak up right now. And just unmute yourself and speak if you would like. All right, I just wanted to give people that opportunity. And of course, if you think of something at any time, we always would love to hear from you. So just give us an email and let us know what you're thinking. Okay, so the first tool I wanted to go over is called the Signal Sifter. It's under the Tools menu. And I'm in the Common Metabolic Diseases Knowledge Portal right now, but this is available through all the knowledge portals under the Tools menu. So the purpose of this tool is outlined in this graphic. It can just let you find variants that have a desired pattern of associations for multiple phenotypes, and it's very versatile, and you can really specify a custom criteria to find the phenotypes that you like. And I'll just give an example here. Let's see. Let's start with our favorite phenotype, type 2 diabetes. And when you select the first phenotype, you get this graph, which shows these are clumps of variants. So variants are clumped by linkage disequilibrium to kind of simplify the presentation. So in each of these clumps, there are multiple variants that are all inherited together. And the variant you're seeing here is the top, the most significantly associated variant in that set. Now, suppose we're looking for, we just want to restrict this to the genome-wide significant variant, so we can put in a p-value threshold, put in genome-wide significance. And then we can also specify whether we want variants with positive or negative effects. And I'll put positive. So these are variants that significantly increase the risk of type 2 diabetes. Now we can put in another phenotype, and let's put in BMI. Now we see on the plot and in the table the clumps that have significant associations for both T2D and BMI. And this is, so for example, in this clump, which has the significant variant for type 2 diabetes, there's another variant that is significant for BMI. So it's not the same variant. Like in this case, for example, in this highlighted row, it is the same variant. And so this is, I guess, colocalized. This is the not rigorous colocalization, I suppose, where you're just looking at variants that are physically in proximity to each other. But again, we can specify parameters here. So let's just let's specify genome-wide significance. And let's now look at variants that have a negative effect on BMI. That's kind of paradoxical, that the same variant, or sorry, I've been sitting still too long and the lights go out automatically in this room. Okay, so we've now specified that we filtered it down to show the variants that increase the risk of type 2 diabetes at genome-wide significance, and they decrease the body mass index, also genome-wide significance. And you can just keep on adding phenotypes of interest. Let's look at coronary artery disease, since that's related to these phenotypes as well. We can do that too. We can see if we filter it down even more. Will there be, yeah, so now we're down to just a few variants. But so you can see we've got a set of variants here. The blue indicates genome-wide significance. So we've got a set of variants that that are very significant for all these phenotypes and in the directions that we specified. I see we have a suggestion from Rani. Suggest you implement a formal test of pleiotropy to rule out co-localized results driven by trait coheritability. Could you say more about that, Rani?
Sorry, I missed the last thing you said. What did you say?
Oh, I just saw your comment, and I wonder if you could just elaborate on that. I'm
Yeah,
not sure
so
I understand it.
if two traits are correlated to a shared genetic architecture, what you're seeing just might be due to that rather than actually a co-localized, as you call it, a co-localized signal. So what you'd want to do is actually implement a formal test of pleiotropy that accounts for that correlation. And thank you. And then wait, sorry. Yes. And then what you would have to do is just test for that, and then you would know if that signal is beyond what you would expect by chance. Because if two traits are correlated, you actually would expect some signals to maybe potentially overlap, and that's not truly a co-localization, that's just a shared genetic architecture.
Okay, yeah, and this isn't meant to be clear, this isn't meant to be real co-localization, but it's kind of gives you a hint that something might be going on. Thanks, that's really helpful. Okay, so that is the general outline of this tool. Doug has a question. Go ahead, Doug.
Hey, Maria, I might have missed it. What's the definition of a clump?
It is, oh gosh, let's see.
Is it a
On
physical
our-
position range of physical?
I think it's a range of R squared and LD, but is there anyone in our team who can give a better answer to that on here? The formal definition of LD.
Maybe there's a threshold that was used
I
to,
think it's
if you're
just
in the clump or not in the clump?
that's my impression, that it's R squared threshold.
Okay.
I cannot give a precise answer to that, but I'll get back to you on that. I'm just making a note for myself. Okay. Thanks for that. I'll get back to you. Okay, any other questions or comments about this one? Okay, let's look at another tool that you may not know of, and this is an analogous, sort of analogous tool, but this is for genes. So it's based on it's the same sort of principle. You choose a phenotype, and then you can add multiple phenotypes, and you first gather the genes that are associated with those phenotypes by their common variant association. So by MAGMA, the MAGMA algorithm takes common variant associations and generates a gene level score. But then this interface allows you to add and filter by a lot more different kinds of information. So I'll just go over an example of that. Let's do the same thing that we did for with the Signal Sifter. So we'll start with type 2 diabetes. And it's just loading right now. Okay, so when you first just enter a phenotype, you'll get a list of genes that are associated by their common variant associations. But you see, the table has a lot more information in it as well. Let's go ahead and add another phenotype, and then I'll go through what else is in the table. So let's add BMI again. Oops, I have to scroll down a little bit here. All right, so we've added T2D and BMI. It's going to reload. Okay, this is a little slower than usual. There we go. Okay, so in the results table, we now have per gene, we show the p-values of the gene level scores from common variants for each trait. We're also showing the huge scores. So these are based on the Human Genetic Evidence Calculator. It's a method that we developed here in our group that takes both common variant and rare variant associations into account and generates a level of evidence for the involvement of a gene in a disease or trait. So here, the huge score is very strong for TCF7L2 to be involved both in T2D and BMI. Also in this table, if there are rare variant associations for this gene, and we can generate a rare variant gene level association score, it would be shown here. You can see that some of the genes have this, but we don't have this for all genes and all phenotypes. There's a link to view this in the variant sifter interface, which I've shown many times before, and I'm not going to show it today, but that'll give you a regional plot of the gene and let you explore other types of information. So this table is, so the graphic gives you a quick overview of what's in the table. So the common variant p-value is shown by a gray circle, and the bigger the circle, the more significant the p-value. So you can see right away which ones are significant, and also that the darker green background, the higher the huge score is. So like this has an extreme or compelling rather evidence that this gene is involved in BMI. But let's add some more information. Let me show you the types of information that we can add. So I think you all know that we are curating from the literature predictions of effector genes for various diseases and traits that a lot of studies are doing these days. So it's the set of genes that are predicted by various types of evidence to be causal for a trait or a disease. So let's add some effector gene lists here. Let's add, here's one from the cardiogram consortium for coronary artery disease. So if any of these genes are on this list, that will be indicated here. And you can see that some of these genes are on this one list for coronary artery disease effectors. Let's add a couple more heart-related traits. Let's add heart failure. We have two different lists for heart failure from two different groups. So why don't we add them both? And now we can see that which genes are implicated by those groups. And let's see. Okay, so we can also filter the results by a lot of criteria. So, for example, the huge score is a measure of significance of association of the gene with these traits. So let's put in 40 as a threshold for the huge score. That's whoops, sorry, that is very strong or better. So now we've got a list of genes that are very strongly associated with both T2D and BMI. And I meant to mention, in the previous tool, the Signal Sifter, I was able to specify direction of effect, but Magma doesn't give us a direction of effect for these gene level scores. So that's why we're not looking at that here. Let's see, we can also look at tissue-specific gene expression for these genes. So let's just put in adipose tissue, or no, let's put in cardiovascular system. And now in the table, we also, wait a minute, sorry, tissue cardiovascular system. There it is, okay. Now we see the gene expression in the cardiovascular system, and these numbers are transcripts per million, or TPM, and sample number for this tissue. So we can also filter by this. Let's say we want the TPM to be at least 10 or above. So these genes are more strongly expressed in the cardiovascular system. So you can see that we're compiling a list here of genes that with really specific criteria that are associated. We can look for genes that are expressed in certain tissues and associated with certain phenotypes. We can keep on adding multiple tissues, multiple phenotypes. We can also, yeah, I think I'm not sure I filtered by the just the common variant p-value, but that would be good, I guess, to add. We can just keep on filtering this list until we narrow down to a really strongly implicated set of genes for whatever we're interested in. So, and you can download this whole table too. So this is just a way of amassing a set of genes that you may be interested in. Doug?
Going back to that p-val rare again,
Yes.
could you remind where that it comes from? Sequencing data
Yeah.
that used a gene-based aggregate test where you bundle the rare variants, or could you just
Exactly.
elaborate on that?
Exactly. Yeah, let me show you on the gene page where that is shown. So that most of them come from a study, an exome sequencing study that was done for diabetes and some cardiovascular phenotypes. I mean, cardiometabolic phenotypes. So here on the gene page, if you look at the rare variant associations tab, here you see. So these are the phenotypes that we have. Oh, actually, okay, that's more than. Yeah, that's right. We've got them from a couple studies, sorry. But so yes, they're exome or whole genome sequencing studies that go into this, and the burden of association, the gene level score is calculated by aggregation tests. Does that answer your question?
Yeah, I assume the ones that have the dash there would, over time, you would have more data coming in
Yeah.
from sequencing, which makes me ask, and you anticipate, or is that a work in progress now, in taking some sequencing, whole genome sequencing?
Yeah, we would like to. Yeah, I don't know that we have a particular data set in mind or in the queue at this moment, but that's definitely a goal.
Great.
Other questions or comments about this interface? Okay, and any general questions about other tools or about the portal in general? All right, well, I'm gonna move on then to just quickly show you or talk a little bit about a tool that we have in development that I think is going to be really, really interesting. Whoops, are you seeing the wrong
We're seeing the presenter view.
center view? Okay. Does that look better?
That looks great.
Okay. Okay, so I've showed the variant sifter in this venue many times, and I'm not going to go through the mechanics of the interface today, but basically the workflow is that you can start by choosing a phenotype and a genomic region to look at, you know, centered on a gene of interest usually. And at first you get a plot and a table that shows all the significant associations for that phenotype across the region. And then as you step through the interface, you can add criteria. For example, credible sets for that phenotype, epigenomic annotations of various kinds and in various tissues, and variant gene links for connections that have been established by 3D chromatin conformation studies between variants and gene promoters. And so you can select multiples of these. You can select multiple phenotypes, multiple annotations, and so forth. And every time you do, the table of variants that initially showed all the significant variants across the region filters down. So at the end, you get a table that just shows the variants that answer all of the criteria that you specified, basically. And the next iteration of this, which is in development, is I think going to be called the Knowledge Portal Signal Sifter or something similar. I don't know if we've decided on a final name yet, but in this one, you'll choose a phenotype to start. And then instead of just seeing a region, you will see the whole genome. So you'll get a Manhattan-like plot. These are not individual variants. These are LD clumped variant associations across the region, so across each chromosome. And so this will allow you to drill down from this Manhattan plot to individual variant associations in any region of interest. So this is very different from anything. We do not have an interactive Manhattan-like plot at the moment. So that's a big difference, a big change. And the other great thing about this interface is that it will basically allow you to access and download all the data that we have in an easy way. I don't think we have every single piece of data that, like, I'm not sure that it currently includes huge scores, for example, but it certainly includes associations, credible sets, annotations, all the, all the major types of information that you're used to seeing in the knowledge portals. So in this interface, there will be different sections for all of these types of knowledge, and you can generate tables of them, and you can compare variants with each other, genes with each other, and so forth. It's going to be very, very versatile. Another change is that we currently show global enrichments at the tissue level. So global enrichment, which is calculated by LD score regression, is when you look at whether genetic associations for a phenotype are enriched within the regions that are annotated as say, enhancers in a given tissue. And that type of enrichment can suggest which tissues are most important in which phenotypes and diseases. So previously, this has only been possible. We've only offered these values for tissue groups like adipose tissue. But now they will be available at the biosample level as well. So like a particular cell type that is within the adipose tissue group, you'll be able to see the enrichment there too. And finally, it's going to have much improved visualizations and user experience. It's going to be easier to work with, easier to select just what you want to see and see it more easily. So it's going to be great. But it's not really ready to show the interface at the moment yet, so I'm not going to demo it today. But I would love to hear any comments on that functionality or other functionalities you would like to add to that.
Will this supplant the Signal Sifter you just showed us because it's got more information in it?
I don't know that we've talked about that. And maybe I think it's DK. Yeah, DK, maybe you have talked about it. DK is our developer who's working on this, but I could see the value in keeping a simpler interface that just does one thing and a more complicated.
Yeah, so this new Signal Sifter will be central place that you can skim all the information through quickly, and then when you need to find out more detail or more narrow down or whatever information, you can go to these individual tools and then find out more on the subject you are interested in. So we are not going to actually throw away all the existing great tools.
Great, thanks. Other questions or comments? Okay, I don't want to put you on the spot, but we really honestly would like to know how you use the portal, what kinds of questions you ask, what other tools you would need. So please do get in touch with us and let us know those things, because we really we want to serve our community. That's our mission. And I guess with that, we'll end a little bit early. Thank you for attending, everyone.
Yeah, I was gonna say, I just want to go over some.
Oh, I'm sorry,
upcoming-
Mackenzie, I forgot. Yeah, don't leave yet.
We just have some upcoming workshops that we'd like to highlight. So first up, we have the Festival of Genomics and Biodata, which will be in London on Wednesday, January 24th. And this event is at the Genome Dome, and we'll be workshopping on the Biomedical Knowledge Portal Network. And then we have the Korean Genome Organization Winter Symposium, which will be Tuesday, January 30th, from 7:30 p.m. to 10:30 p.m. Eastern. So this is an international workshop focused on the Association of Function Knowledge Portal. So if you're available for either of these, we'd love to have you participate and listen. And just some heads up for our schedule for 2024. This is the schedule for the Knowledge Portal webinars and focus groups. I did look at the calendar, and it appears that I do need to add some of these to the actual calendar itself, but this is what we have scheduled. So we really look forward to 2024 and having more opportunities with everyone. Thank you all for attending today.
Thanks, Mackenzie. And thanks to everybody. Bye-bye.
Bye.